So a good cardinal rule in life is to always leave a place better than you found it. So if you're ever teaching, erase the boards when you're done. It works in just about any situation. Stick to that one, you'll always be good. All right, how you guys doing? You guys ready to get rolling? No. What do we need to do to get rolling? What do we need to further prepare for? I'm sorry? More coffee. Okay. Well, I'm sorry. I can't help you there. Yes, ma'am.
As far as like the quizzes that have you been doing on Blackboard? I can't find them. There's just one, but like of the syllabus. Oh, just that one?
Yeah, there's just one practice one.
Oh, okay.
It's just to show you how the system's going to work.
Oh, okay. I thought it was going to be part of it.
No, so the ones for the practice test are noted in the syllabus for the actual textbook's webpage.
Oh, okay.
So you got to go to just, if you read the syllabus, and it'll say that there's, you know, I think it's Mind Machine 2E or something like that. I'm sorry?
Is it the book?
Yeah. So that's what it says in the textbook. Not just the study guidebook, but there's a website that has practice quizzes.
Oh, okay.
So if you just read basically the instructions on the syllabus and then follow the instructions in the book, either way, I think it should work.
Okay.
And if it doesn't, just let me know. Shoot me an email. So that should work. There is a practice quiz that is basically on Blackboard. It's not about content. It's not about what ventricle connects to which other ventricle, or it's not about that stuff. It's about 30 of you guys have taken a look at it, it seems. So you guys already know that. Obviously, you should be familiarized with what's in Blackboard and updates on it, so hopefully you guys saw that. It's to give you some familiarity with how the system will be for taking the exam next class. Okay, so please familiarize yourself with that so you know what's coming, because we're not going to have time to BS once class starts on Thursday. We're going to dive right into the test. Since we've talked about it since day one that it's going to be on Blackboard, you're going to have your digital systems up and running and connected to the internet and all that. All that's expected to be done. All right, so I'm not going to have time to walk around to every single person and get your internet connected and log into Blackboard. That's all expected that you guys are bringing to the game. Okay, so that's why that quiz is there, is to help gain familiarity with exactly how this is going to work. Sound good?
So the test is this Thursday?
Yes.
I noticed on the Blackboard, we're able to go back. Is that, we're not going to be able to go back?
Yeah, right now we're not going to be able to go back on it. So, know the answer? Be sure. Okay? Just like your GREs, just like your MCATs, your LSATs, any other kind of SAT of a test that you might have to take afterwards, we want to practice getting prepared for that model that the field is moving towards. So we'll do that. A couple of people have asked a couple other details about it. Of course, that practice quiz, or whatever that's on there, it obviously says practice quiz, and obviously the syllabus doesn't say anything about grading for that element into the grade for the class. So no, that's not going to count to your grade if you get one or two questions wrong or right. Don't sweat it. It's not going to affect you one way or another. It's there for what Allen Iverson said, is practice. Okay? And likewise, it is important to spell things correct. You know, if there's a slight variance here, basically I'm going to kind of just use a common sense approach to that. If it's very clear that you know what you're talking about and there's like one letter wrong and nowhere epinephrine or something, you know, it's an accident. That's, not going to be a spelling Nazi to you. But that being said, it's important in college to spell things correctly. Okay? Does that sound like a pretty fair deal? Okay, cool. So, speaking of norepinephrine, one of the things we raced through at the end of the last class, because I begged your indulgence for the last 2 minutes, if you might remember, was we talked a little about some neurotransmitters. So hopefully you guys picked up the cue that I mentioned back then, which was to focus on certain elements in Chapter 4, not necessarily the entirety of it, because we're not going to get into all the drugs of abuse and things like that on the exam. But I did want to just remind you guys of what it was that we referred to in terms of the neurotransmitter systems that are going to help us to provide a base for understanding other elements of the class in the same way that the neuroanatomy, the orientations do. Okay, so we talked about basically there being 4 different types of neurotransmitters that we can categorize them in different classes. Okay, so the first one might be amino acids. Okay, and there's 2 big players in the game here. Basically GABA and glutamate. Now GABA is this nice long word, GABA aminobutyric acid. You don't need to know that. You're not going to have to spell that. Everybody in the field just calls it GABA. Okay? We can contrast that to glutamate in the context of which is excitatory and which is inhibitory. Okay? Now we'll get more into the elements of what excitation and inhibition means after the test and the next lecture. Okay? But know basically that glutamate is excitatory, GABA is inhibitory, which means glutamate is going to basically make the brain systems a little bit more excitable or a little bit more active. GABA is going to kind of tamp it down a little bit. Okay? So basic elements there. Okay? Glutamate works like we talked about on these different receptors called AMPA and NMDA. There's also a third one, but we're not going to get into that for this class. And the GABA receptors basically allow chloride in. Just as a note, it's one of the targets of classes of drugs like benzodiazepines and other things like that that generally work to kind of relax systems down. So at a very simple level, that's it. Okay? There's also a class of neurotransmitters called peptide neurotransmitters. Okay? So these are basically what you might think of as some of the hormonal ones, oxytocin, vasopressin. We're not going to talk a lot about them, but if you've had other classes here, in like evolutionary psychology or other elements that I know people teach, you might be familiar with those. Okay? There's also this other class of neurotransmitters that we might talk a lot about later, which is more what we call the amine neurotransmitters. And they're basically acetylcholine, dopamine, and serotonin. Okay? If we have time later, maybe I'll show a fun video about the role of acetylcholine, and another one we'll talk about norepinephrine later, in the sympathetic and parasympathetic nervous systems. Okay, remember we talked about those last class? Oh, I should say too that just so you guys know, I'm going to put today's slides up on Blackboard. Okay? So I see a lot of people writing down furtively. I'm not saying don't take notes, but I want to give that element of context so that you can relax about the tiny details if that's what you're trying to copy down. Okay. Anyways, so we have basically things called acetylcholine, dopamine, and serotonin. All right. We're going to talk about the role that serotonin can play in psychopathologies like depression, dopamine in the context of certain psychopathologies, context of schizophrenia, also learning and memory. Acetylcholine is also very involved in learning and memory as well as peripheral systems too. All right, and just briefly we'll touch upon some gas neurotransmitters. Basically, transmitters that go almost basically backwards from postsynaptic to presynaptic sides of the synapse. And we'll talk about that briefly, particularly when we talk about plasticity and learning and memory towards the end of the class. Okay, so just to kind of show you those systems, this is kind of what we finished off with last class. We showed this really quick, and I'll just show you kind of briefly the elements that I think it might be important to focus on. Okay, so just when you see some of these abbreviations too, so serotonin is typically referred to by a chemical abbreviation, 5-HT. Okay, norepinephrine is basically shown by NE. Dopamine is abbreviated by DA, and there's lots of different receptors for dopamine too. So you might see the dopamine 1, the dopamine 2 receptors. Okay, but DA, and then acetylcholine is generally abbreviated as ACH. Okay, we'll talk about these brain systems in more detail as we get along. Okay, so acetylcholine, ACH. Okay, basically they're synthesized oftentimes. All these little transmitters we'll talk about, they're synthesized in special places in the brain, and then they're projected all over. Okay, so this is one of the ideas or this is one of the reasons that underscores the idea that And the challenge in treating certain psychopathologies, treating certain mental disorders with certain pharmacological approaches, because let's say you know that something like serotonin is deficient. Well, fine. Or acetylcholine. Well, we know acetylcholine is basically maybe synthesized and projected from the basal forebrain area, okay? And it projects everywhere else, okay? So maybe there's a problem in one of these little elements, but if you, let's say, increase acetylcholine, what it's going to do is it's going to increase all these other areas of the brain that are going to be responsive more to that particular chemical. Okay? Well, maybe the only problem was that it wasn't working in this particular area. All these areas were fine. All right? It's a real challenge with therapeutics and pharmacology, right? Because what you're basically doing at that level with like those chemical drugs is you're basically attacking the problem with a shotgun instead of a rifle, right? What you want to do is pinpoint one shot for what you would need, just on target, as opposed to a shotgun that's just going to spray everywhere. And sure, you might hit the target, but you're going to hit a lot of other stuff around that might get damaged. That's not what we want to do, right? But it tends to be what sometimes we have to do pharmacologically. So some elements to keep in mind as we move through the class, okay? Dopamine, okay? The key areas where it's synthesized and projected from, okay, are two. One, what's called the mesostriatal pathway, which is basically the striatum. Striatum, remember we talked about the basal ganglia, okay? So sort of that middle element of it, and basically going to go from the substantia nigra up to the basal ganglia. All right. Now it also has this projection, okay, to the what we call the ventral tegmental area, okay, and that's going to go to the nucleus accumbens. These are midbrain areas. Nucleus accumbens in the midbrain, okay. So just a couple areas to know that are associated with synthesizing and producing a lot of these chemicals, and then they project to all sorts of areas in the brain, and they do all sorts of things. Dopamine in particular is heavily involved in learning and memory, as well as movement and motor control, okay? And so we'll see elements of that moving through the course. All right, serotonin, its neurons that we will refer to as serotonergic neurons, okay, tend to originate a lot of the synthesis for this chemical in an area called the raphe nucleus, okay? Here, raphe nucleus, okay? It's a lot of brainstem areas, okay? So it's going to come both project downwards and then throughout these cortical areas too. So you notice a lot of these pictures, they all show a lot of these same sort of cortical projections. They're very broad, generic cartoon arrows, right? That's on purpose because it's accurate. There are, you know, major projections to millions of different neural sub-areas, and that's a generic, accurate way to represent those projections, okay? So we talked about these serotonergic neurons, the raphe nuclei, and the brainstem, and then we can also identify the place where norepinephrine is synthesized and projected from, which is an area called the locus ceruleus, okay? That's a kind of a crazy word to see and spell and pronounce, okay? But good practice there will help. It's pronounced as ceruleus, okay? And a lot of its projections are going to go to the lateral tegmental area, and it's going to be involved in things like attention, mood, sexual behavior, those types of alertness, heavily involved in those models of attention, okay? So that's kind of the idea there. We mentioned the idea of gas neurotransmitters, and the idea here is that if you have a presynaptic neuron and a postsynaptic dendritic spine, okay? A lot of times our neurotransmitters, right, they're released in that direction. All right, we've talked about this. All right, a lot of the gas neurotransmitters operate as retrograde signalers. All right, so they're kind of like the hipsters of the neural signaling world, okay, and that they're going backwards in time. So a lot of you might have these gas neurotransmitters, and they might come and operate from postsynaptic to free. Okay, and we'll look at some mechanisms of how that happens as we move through the course. All right, but I just want to introduce some of these elements so we have a basic idea of how that different elements can work. Okay, yes?
Is that like the recycling effect of serotonin goes through and not all of it's used, it goes back up?
No, that's a little bit different. So, remind me your name? Amanda. So what Amanda had asked, if anybody didn't hear, what she asked, is this the same? This idea of a retrograde signal, or is this the same as what she heard about, essentially of like a recycling of neurotransmitters? For an example, from release of serotonin, when serotonin is sort of done or too much being released here, will it be recycled back into the presynaptic cell? And that's a little bit different, because what that is basically doing, you're reuptaking on the presynaptic side what's released here and is sort of coming back home. Here you have it leaving from a different destination entirely, and it's operating physically as a gas substance as opposed to a sort of a traditional neurotransmitter chemical. So that's kind of the real big difference there, but good way to think about it. Okay, so basic chemicals that help to communicate information in the brain. Okay, norepinephrine, dopamine, serotonin, acetylcholine, GABA, glutamate. Okay, glutamate, one of the chemicals in MSG. How many of you guys like ramen noodles? I do. MSG, monosodium glutamate. Glutamate in there. Okay, it tastes delicious. Okay. Anyways, so we will talk at length in the next lecture, a week from today, and next week, about the electrical properties that allow these chemicals to be released presynaptically to postsynaptically and allow these signals to happen. Okay, so we'll talk about the electricity, okay, going in there and how that works in the next time. But today I just want to introduce the chemicals that mediate this neural communication, what you might call neurotransmission of information between different neurons. Okay, so we'll pivot to now what I want to talk a lot about today is a little bit more depth than what we've covered so far on the methods of how we measure the brain. Okay, so if you can imagine that you have this big like cantaloupe in your hand, okay, and you're not allowed to crack it open. Okay, maybe it's even more like a coconut where it has a really hard shell on the outside. And you're really thirsty on a desert island. So you really kind of want to know what's in there and kind of get at it, right? But you're not allowed to crack it open. How do you know what's in there? How do you know what's inside of that? Okay, so you can shake it. Okay, what else? Yes. You can weigh it. Yes. Tap it, shake it. So you can do all these caveman things. Right? Yeah.
You could do like an X-ray or like a MRI.
Well, you could do an X-ray or MRI of a cantaloupe. Yes, you could. But let's imagine that we're on this desert island and we're not quite as close to the local X-ray facility as we might imagine. Yes.
You can try to move it around to see if you hear anything.
Okay, so shake it. So move it around, see if you hear anything. Yeah.
Spin it.
Okay, so you can spin it too, and then do what?
Well, spin it will tell you whether it's solid or liquid inside. Because if it's solid, it'll spin upright. If it's liquid, it'll fall to the side.
Okay, so now we can actually start doing some manipulations. You spin it one way, you can see if liquid spins around or if it falls down. You can start basically doing manipulations that allow us to make observations that permit us then to deduce certain ideas about how it might be.
Right.
You still might not know for sure. And all the example is that I try to use to communicate is that this is really freaking hard, right? Because we're basically reduced to caveman-style approaches to trying to understand what's in that cantaloupe or that coconut, because it's hard, right? Well, that's what our job is as neuroscientists. As psychologists, we're trying to figure out what's going on in this big coconut on our neck, okay? It's really hard because, for the most part, we're not allowed to sort of take a saw and a drill and open up and go inside and peek. We'll talk about some of the ethical considerations why that's not necessarily considered a good idea, and some ways that we typically find as ethical to also get around those limitations. However, the idea is that it's a hard task. It's a big puzzle and a challenge. And we're all trying to figure it out in the whole field. And so we're going to talk about some of the methods that we use to do that. And some of them take some of these approaches and integrate them with some aspects of technology. Okay? So keep that in mind. As we talk about these things, none of them's perfect, and they're all basically ways to try to get a snapshot of what we think might be happening. And none of them are perfect, and we'll talk about some of the pros, cons, advantages, disadvantages, and all that kind of good jazz. Okay, so we've talked about Golgi stains before. How much of a given tissue does that typically stain?
5%.
Okay, about 5%. And what features or morphologies of the cells does the Golgi stain tend to stain?
All of it.
Oh, yeah, the whole cell. The soma, the projections, dendrites, axons, so you can see everything. Okay, and we talked about earlier in class the idea that sometimes there's a trade-off, and you might want to see other stuff that you would sacrifice other abilities to visualize. Okay, so we talked about Nissl stains where you're only going to see the cell bodies and not the projections, but yet you can see all of the cell bodies. Okay, so you have this trade-off. Okay, some advantages, disadvantages, we've covered it already. All right, you're identifying where is that RNA located, because RNA is going to be situated in the nucleus. Okay, it's a very useful way to image at a cellular level, but also in sort of the gross anatomy too. You can see all these cell bodies in the gray matter. Now, of course, it shows up purple in the Nissl stain. All right, we talked about autoradiography, where you can see where there is activity. So now you're not just looking at structure, you're looking at function, stuff that's active and stuff that's not active. All right, now we've seen this picture before, but we saw it in black and white. It's a flattened piece of tissue from the occipital cortex. You guys remember where that is, right? Yeah, all the way in the back. Okay, that was active when a certain, I guess it was a monkey, was looking at a grid on a computer screen that basically looked very similar to this. Okay, and the idea here is that by transforming that image, we can see that where the autoradiography stained this tissue corresponds really nicely with what was seen here. Okay, so we can see where things are active, and we're going to maintain this idea of comparing the ability to image, which just means where can we see, because we're trying to see what's inside that coconut, right? We're going to try to maintain the idea of contrasting the concept of imaging structure versus function. So some methods, like Golgi or Nissl, allow us to see really good structure, but very, very little about function. Okay? Things like radiography do allow us to see some elements of function now, but what does it show us for structure? Well, it shows us where it's active, but it shows us basically black dots. Nowhere near the elegant structure that we can see some of these other approaches. So we're just contrast this stuff as we move along. Okay? We mentioned immunocytochemistry earlier in class. Okay? I drew this cartoon up on the board back in the day. All the way back a week or two ago. Okay? You might remember it. Okay, we looked at figures like this, where you can label different cells that have different proteins with different colors. And this is just the way you do it. This is our cell, this is our membrane, and we want to find this particular protein. We know that there's an antibody that really likes it. Really binds with what we call a high affinity. Remember that word? Okay? So there's high affinity binding between this primary antibody. But before you throw this antibody into that animal system, first, you make sure that you tie together a marker to that antibody. We call a secondary antibody that's tagged with a certain fluorescence color. That way, if you tie this stuff all together first, and then you inject it into the animal, you can see where does it bind. Remember we talked about that? So this is here just as another illustration of that. Okay? We also talked about ways that you can tag radioactive sequences of DNA and RNA. So basically when DNA is sort of unzipped into RNA, you can add a radioactive element that will fluoresce under certain conditions, and you can then see where that particular DNA is. Okay? We talked about that. We did not talk about yet this idea of optogenetics. Okay? It's a really complicated tool that we could spend all year talking about. But for the purposes here, just want to introduce the idea that it's a really, really new technique in the field. It's really cool, and what it allows you to do is basically take advantage of ideas take advantage of what we already know about certain proteins that are really sensitive to light. So those proteins and the channels that we talked about here, right? Remember we talked about that? And sometimes we said those channels are closed, sometimes they're open. Sometimes the way that those proteins will open or close is, amazingly enough, dependent upon whether a certain type of light is turned on or off. So you can engineer that protein into cells and then inject something to turn lights on and off, and now you can actually experimentally control what neurons are on and which neurons are off. So if that gene for that protein is integrated, let's say, in a motor center of the brain, and that little mouse is kind of like walking along looking for some cheese, and you turn the light on it, because now you turned off that cell. So how's it going to move? The motor area has the cells turned off. Then turn the light back off, and it's like, okay, we're going to eat some more cheese, or what have you. It's a really new and powerful way to affect brain activity and to manipulate it, as opposed to simply standing back and observing it. Okay, it's a very powerful approach that scientists can use. Okay, it's called optogenetics. So that's one way to do that. Okay, another type of method that's also very similar to the idea of optogenetics, in that it is new, exciting, and still being developed, is called clarity. Okay, it's developed by Carl Dieseroth at Stanford, and the idea is this is a way to image structure. Okay, and it's a way to wash out a lot of the tissues of the brain and image just certain structure. Okay, so this is a mouse brain. Okay, and this is it here. Okay, and you can see it's over this little writing. You can't read what's being read there. What you do is you use some chemicals to wash out all these extra lipids and proteins, and you're left with the structural elements of the brain that now, this is actually, after it's washed out, this is still the brain hanging out here, but you can see straight through it. And you can image it using some different tags that you can inject to see it under fluorescent light, and you can actually see these colors illuminate some of the connectivity in areas of the brain. Okay, so this is integrating some of the methods that we've talked about before. Okay, and so if you guys are interested, I provided some sort of supplemental videos where they actually interview the inventor and the discoverer of these techniques, and you can see a little bit more detail of it on your own. It's really cool. Okay, so anyways.
So
Yes.
you bind together what you want to keep, and then it washes away everything else?
Yep. Yeah, it's really creative, really, really cool.
Acrylamide?
I'm sorry?
Acrylamide, is that what you said?
Acrylamide? Yeah, is that what they have? Yeah, acrylamide. Yeah, so, if any of you guys want to get out in the forefront of the field and kind of like lead the charge in next discovery somewhere, this is an area that you can start to think about and learn about. Okay? Now, if you want to image other structural elements, okay, not so much, say, in a mouse brain, but let's say a human brain, one way to do that is called diffusion tensor imaging, or you can just call it DTI. So this is a way to image the long tracts of axons that we've identified in the brain, like the corpus callosum. You guys remember we talked about the corpus callosum? Okay, so that's what we see here. This corpus callosum. See how it kind of looks like that brain? Sort of looking down from the top view, right? Okay, so maybe you have an anterior, posterior part. These are those connections going across the left hemisphere and the right hemisphere. And one thing that's evident here that you can see but quite clearly is that this is not just an artist's rendering because it looks cool. Okay, the colors have meaning. The colors show the strength and the size of the connections. And in this case, the scale is such that red is the biggest and strongest. And we know that because that's basically the corpus callosum. Okay, now the way the DTI basically works at a conversational level is it takes advantage of something called Brownian motion in the world of physics. And the whole idea is that basically the machine and the technology is able to image. Well, let's walk it back actually. When you're imaging, what you want to see are tracks of axons and where they connect and project in the brain. Okay, remember you got to put your cell phones and stuff away during class. That's policy if any of you guys are using it. But the idea is that when you have these projections that you want to image and see where do they go inside our coconut craniums, right? Because we can't crack it open and see where they go. Okay? These axons, there's water in it, slight water molecules, because it's a cell, right? And water will diffuse in different areas, unless it's wrapped up in myelin. And if the myelin is wrapped up around it, it's not able to diffuse across, it'll only diffuse out and down. Does that make sense? So you can get a way, basically the method is a way of identifying what direction are the water molecules moving in the brain, and anywhere where they're not moving sort of laterally and you can see that they're just going sideways, now you can reconstruct, the computer knows that that must mean it's an axon, at least a myelinated axon. And if you know that, then the computer can start to build that information with, basically algorithms that will then reconstruct it, and you can see based on the strength of that movement of water molecules, what you call Brownian motion, for any of you physicists in the room, it'll tell you where those axons are, and the strength of the movement is going to tell you the thickness, essentially, of the myelin. Thickness of the myelin is going to correspond to the size of the connections. You see the sort of connection there, the logic, no pun intended? It's a really creative approach, and then you can reconstruct it, and you can see sort of the relatively smaller, weaker connections and the larger ones. That's sort of DTI, and it's really useful to provide information of structural connectivity, particularly in humans, which is really tough to do. Okay? Other ways, if you want to see structure in the brain, you can do what's called a CT scan. It used to be called a CAT scan, and I think people just call it CT. It's kind of an older way to take a still frame picture of the brain. And it basically draws upon X-ray technology, and the X-rays will basically give you a still picture kind of like this. And you can see some areas of damage or deficits there. You can go in a typical X-ray machine, and you can use different approaches to basically generate this anatomical map. You inject a dye, you see where it's basically projected to, and you take these different X-rays from different angles moving around, you can reconstruct it. Okay? So you can't tell function though. But it's really useful if you want to see what's hurt or what is where. Structural information. Make sense? Okay. As time went along in the pantheon of modern history of brain imaging tools, particularly for humans, the CT scans were nice, but they didn't have a great resolution. Okay? And what people developed what was called magnetic resonance imaging. So we got a lot more depth and detail that became available by taking advantage of magnetic fields. Okay, and the idea here is that we can get this better resolution, okay, without having to expose people to the risks of X-rays, which is what? Radiation, right? Okay, so you don't really want to do that to people for ethical reasons, because it's not really fair to them, just so that we can get our thesis done. Here, let me juice you up with some radiation. Do you mind? Right? That's not good. So this was a more useful way, and so it tends to be a more used way now, because it has two major advantages over CTs. One, it's a better picture. It's more high res. You can get finer detail, and it's safer. No radiation exposure. So key things to think about. Okay. And the way that it's basically working is taking advantage of magnetic fields. So you can use these magnets to basically cause the protons in the brain to basically align to a certain direction. Okay? And then you use a radio frequency pulse to sort of disrupt that. And you can measure the rate of response back to their original position. And then computer algorithms will reconstruct that differential rate of response of those protons and electrons in the proper directions of their spin, and you're able to basically reconstruct that information into a digital picture. Okay? And the idea behind there is that different tissues have different densities, and so those protons will realign differently in those tissues. Obviously, that's a whole course, so we're not going to get into the details. Don't sweat it. Just know that it uses magnetic approaches to do structure. We still can't tell function yet. All we're doing are taking pictures. We can't tell anything about activation. Okay? So, we go to the next line of development now, which is not structural MRI, but what we call functional MRI. Now we can tell function a little bit. So the difference between basically a picture of the brain versus a movie of the brain. Okay? And still using those magnetic field ideas. Okay? But now what you're also doing is not just detecting basically the return of those protons based on the radio frequency emitted, but you're specifically detecting the flow of blood. Okay, well, why does that matter? I thought we cared about the brain, not the blood. Well, the idea is such like this. If we know the brain needs oxygen, anybody disagree? Okay, be pretty bummed if we cut off our oxygen after a period of time. Okay? We need the oxygen in order for the brain to work. When brain cells work, they use oxygen. When the oxygen is used, those brain cells basically emit and get rid of the used oxygen. Okay? And we can measure what we call basically the blood that carries away, or the blood that is deoxygenated. That's already used the oxygen because it's already just delivered it to that brain area. So, if you can see where in the brain there's blood that is deoxygenated, then you know that area must have just been going on. Because something just used that juice. Does that make sense? So, it's a measure of brain activity, but indirectly. So the key thing to remember is that fMRI is an indirect measure of brain activity because it's actually measuring blood activity. What you might call hemodynamic activity. So what you'll see sometimes in figures and data and papers, you'll hear me talk about it in class, is something called BOLD, okay, which is the abbreviation for blood oxygen level dependent. Blood oxygen level dependent signal. So when we see pictures like this, and we'll see them all over. I mean, you see them in the New York Times, you see them in class, all sorts of different. We talk about areas of the brain that light up, right? This is what we're talking about. They're lighting up, so to speak, showing differential patterns of activity and where they're located in a three-dimensional structural resolution based upon where we identified relatively more blood activity that was deoxygenated. You guys follow?
Did you say indirect measure of brain or blood?
Indirect measure of brain activity. It is a relatively direct measure of blood. But that's why it's an indirect measure of brain. It's kind of like well, yeah. Just keep it simple.
Okay.
Make sense?
Yeah.
Okay, and you know, you can, I don't know, many of you guys might have had MRIs or fMRIs, or just the research or doctor's offices or whatnot. You know, this is oftentimes what an MRI machine will look like. You put them in that bore, it makes a strong magnetic field, it orients those protons, they relax, you measure that differential activity, and you can map it basically together with the blood oxygen level dependent activity, and you can rotate and you can see really cool stuff. Take slices of the brain, okay, and create these differential sort of coronal slices versus the horizontal slices, and you can see areas in a three-dimensional representation that a CT scan won't give you. Okay? And you see the functional activity shown here. We're in a coronal slice like this, seeing which areas are active functioning over time. Okay? So this might be when a stimulus pops up on a screen at time point 0. You can measure, okay, the difference between activity over time, 2 seconds, 4 seconds, 6 seconds, 8 seconds, 10 seconds, between information that's, say, remembered or forgotten, as an example. So it gives us this functional information as well. But here's the thing about the functional information. We're going to add a dimension to start thinking about. Okay? What is the resolution of time that we can detect with this? We're going to call our temporal resolution. Basically, if we're able to study function now in addition to just structure, how quick of time can we measure? Well, in this case, only on the measure of 2-second intervals. Well, the brain is active in milliseconds. We'll talk next week about like millisecond level firings of neurons. So this is really useful, but it also masks a ton of other stuff that's happening differentially. But hey, it's one useful way to get into that problem of trying to understand what's happening in the cranium. All right, so again, trade-offs. Very good structure, reasonable functional activity with a relatively sluggish temporal resolution. Okay? So the thing about too, it's also expensive. It can be maybe $1 million for a machine, another $1 million to maintain it in the facilities and the technicians. Might cost $600 an hour to use the machine when you run a research subject. So when you're designing your theses, you take these factors into account. Okay? You got to have the right tool. You need to use the right tool for the job. Yes.
This is the first time I've heard about it, so I want to make sure I have it right.
Yeah.
I'm also the one that emailed you about it. So it's different than the MRI. Well, it's the same as the MRI, 'cause it still uses the magnetic stuff that's, messing with the protons, and it goes back and they measure that. But this is different because it takes pictures of where the oxygen is deoxygenated.
Yeah. So we're seeing functional information that the MRI will never show you any functional information.
Okay.
It's adding a movie. It's adding a movie on top of the picture.
Because it doesn't measure the oxygen.
Nope. Okay. But some of the advantages, it doesn't use radioactivity and all that kind of good jazz. So, some information there. Now, again, here's your answer to your question. Oh, yes.
For the advantages and disadvantages, is it proof of the thing that I don't see? Because it says,
Can you speak up?
there's advantages and disadvantages, it's contradicting.
In which regard?
The fixed value, solution and speed.
Sorry?
Poor spatial.
Oh, yeah, so disadvantages, it should say basically poor temporal resolution. Good catch.
Thank you.
Yeah. And of course, anytime we're making a qualitative sort of value judgment about what's good and bad, right, you should always have that little sort of word in parentheses that's relatively good spatial resolution or relatively bad spatial resolution, right? So just keep that in mind, yes.
And same thing, if the advantage is it has good speed, but the disadvantage is very slow. What do you mean by that?
Okay, so this is what I mean. Good catch again. What I mean by that is sort of exactly what I just said about the relative aspect. So it has better speed than what we'll talk about, say, PET scans have, but it's also slower speed than what we'll talk about, say, EEG has. So it's a relative issue. So it has relatively good speed compared to some methods, but also relatively bad speed compared to other methods. So, yeah, that's tricky, but it's an important point because it's like beauty, right? Like some things are beautiful compared to some other things, while at the same time maybe not being so beautiful compared to other things. And then, of course, it's in the eye of the beholder and all that kind of good jazz, right? But we can see that it can be both, good and bad, sort of relative to comparison. Yes?
So for the MRI and the MRI, do they have to use two separate machines?
Nope.
It's the same machine?
Same machine. Yep. Okay. So, MRI, again, is using structure. So, this would be sort of just looking at the structure of the brain. Okay. Functional MRI is looking at function, so we're able to sort of identify what areas are active during different things, like looking at donuts, or thinking about sleeping, or sex, or sweet, sweet beer, or TV time, right? We can now start to categorize the functional contributions that these anatomical areas might be involved in. Okay? Does this slide help to answer your question?
Mm-hmm.
Okay. Homer Simpson's always good for that. Okay, so we mentioned PET scanning a little bit ago, okay? This was sort of historically and developmentally sort of in between the development of CT scans and MRI scans, which puts that relative element into your answers to the question. So it's a way of also imaging both structure and function, okay? However, it has some relative advantages and disadvantages too. So the advantages is that now instead of just a structural image that, say, CT scans can give us, this can now image some functional activity,
Okay, so we can change or track the different changes in certain activity in the brain, and it's relatively fast compared to, let's say, CT scan, which is 0. Okay, but otherwise it's relatively slow. And in fact, what I would do is completely X out that, because that we don't even need to think of as being fast. We can just think about PET scans as always being slow. Okay, so I'll try to simplify that there for you. Okay, it's similarly expensive. Okay, yeah, see, it's actually relatively slow. Right? You should be a good politician. You can say 2 things on the same slide and both be right. So the idea here, don't worry about fast, you can get rid of that. It is relatively slow in time. Okay. The way that it can work is you basically inject radioactive substances into the bloodstream, like chemicals like technetium, for example. I had one when I was in high school. They shot me up with technetium or something, and it will basically image where it binds to different receptors in the brain. So it's one way to see where, say, cocaine is binding or something like that, if you're interested in studying drug abuse. It's also areas you can see that are relatively active or less active. In this case, patients who have Alzheimer's disease versus those who are normal, you can see a lot more activation, much lower ones in Alzheimer's. And we'll talk about some of the structural reasons why as we move deeper into the class. Okay? So it's basically using radioactive emissions, and it's relatively slow. So as you might imagine, time has since passed by the PET approach because it's a little bit outdated. We have better methods to see both structure and function without the risks of radiation. Seems like a good deal, and that's why you see so much fMRI. What else do we say about PET? One of the advantages, though, to it that it can do is it can basically tag for, you can use these radioactive tags to identify basically ligands or receptors. So if you want to see like serotonin receptors, where are they located in the brain of humans? This is one way to do it, but it's going to come with a risk. Right? You got to ask your people to sign up and sign consent forms saying they're okay to be shot up with radioactive. You know, it's not going to kill them necessarily, of course not, but it's a lot riskier than sitting here in class. That's all. Just something to think about. Make sense? Pros, cons? Okay. So, the way that the ideas then move forward from fMRI and PET scans and what we'll talk about with EEG and what we'll see as an MEG as well, and how is this useful to identify functional activity? Well, the answer is called subtraction logic. Okay? It's from a guy named Donders in the 1800s. He developed this idea of subtraction logic. And the idea is very simple. Okay? You're imaging brain activity. Okay? Let's say you see one picture of brain activity here and one picture of brain activity here. Well, what was it brain activity for? Well, it turns out one of these was brain activity for when they saw a checkerboard on the screen. Oh, so the visual system was seeing stuff. Okay, what was this brain activity for? Well, it was for basically nothing. So what you can do is take a picture of this and take a picture of that, you subtract them, and anything where they differ, see how they're basically the same up here? There's a middle blurb, left blurb, right blurb, that's all the same, right? This is a little bit deeper red, and this is a little bit lighter red. So when you subtract that, because it's all just numbers being transposed onto colors, right? You're just subtracting the numbers in the matrix that's creating this color map. Anything that differs by virtue of that subtraction is telling you information that you can deduce. Oh, that area of the brain is especially active during this particular stimulus. Make sense? It's the whole power of these techniques is using this approach called subtraction logic. And you get this difference image, and that image is during one stimulation. Well, one thing is just one thing. Who freaking cares? Right? What scientists have to do, because there's variance involved and things are different, you want to see the average over time, or the average of multiple instances, and the average of those multiple instances over multiple different people to try to understand what is true about what areas are active in supporting seeing a checkerboard, for example. So you do that over many, many, many times. You get the average of that difference image of the subtraction logic. And that's the idea. Okay? So that's the strategy.
Is that just those two, or is that multiple different stimuli?
Just those two what?
You have control, and then you have one visual
Yeah.
stimuli. Are you talking multiple stimuli?
Yeah,
or
that's
the
what this
average
is.
of just that stimuli
This-
multiple times?
This is the, well, in this case, let's say it could be this stimuli multiple times,
Okay, all right.
for example, but you could do it either way. That's the idea. This is one here, and then you have another one, another one, another one, another one, another one. Notice they're all different, right? Because the brain is going to be slightly different every time. They're all different. Look at the red spots. They're all in different places and different intensities. We average it over time, and you get the average. You know, it's not perfect, but it's what we use. It's what we have. Okay. And so when you do that, you can create different averages that you can see. Say Alzheimer's patients might have a lot more activity in one condition versus controls that don't. Maybe they'll do something that controls find is really easy. It's a real challenge though for Alzheimer's patients, for example. Okay. And you can just map that, and that's what these colors are. It's not lights up in the brain. It's a statistical map of where things differ on that subtraction. And it just corresponds to a color scale of a t-test. So when you graduate and you're scientific citizens in the world and you forget everything else from class, remember that when you see these fancy, colorful pictures. They're just basically statistical maps. Okay? So that's the idea. And when you see those statistical maps, like all these nice fun words, remember that there's caveats to keep in mind. Okay? There are limitations. Okay? So especially the New York Times loves to bastardize these findings and make them really cool and sexy and exciting. And they're not always right. So you gotta be careful and cautious scientific consumers. Okay? So they had an op-ed that said a few years ago, you love your iPhone, literally. Although that's not actually the right word, the right use of the word literally. Don't you guys hate when people do that and they say literally about everything and it's not actually literal?
Yeah.
Yeah. So they were doing that here, and they did, basically, they reported some finding of some scans that showed an area of the brain that was active in other studies involving like romantic love, say the amygdala, also was active when people saw pictures of their iPhone or something like that. So, of course, you cannot be allowed to make that conclusion to jump that both mean the same thing, but they did. And so you'll get situations like this where usually about once a year or so, there's like 200 neuroscientists that have to write a collectively signed op-ed back to the New York Times telling them that they're wrong and they need to get their stuff together and quit making bad science reports. And it happens every now and then. So you'll see that. Here's some of those areas, the insula that was activated during the iPhone use. Okay. You can see brain scans can be used to reveal what dogs really think of us and all sorts of fun, engaging topics and findings. Okay. But keep in mind that it's all ones and zeros. Okay. Ones and zeros meaning the numbers that are used to subtract what's active versus what's not. Okay? We can see nice images, and they're beautiful, but they're all statistical maps of just what's reliably different, which is useful. I'm not bashing it necessarily. I'm just saying that we need healthy, responsible scientific understanding of what we're looking at and what we're not. We need to know what it means, but not be trapped into jumping to a conclusion of something it might not be. Okay? So there's a couple good ways that people have categorized ways to do that. So if you want to see more details on that, there's a book called Brainwash, The Seductive Appeal of Mindless Neuroscience. Okay? And they'll explain so much more fun details of ways to be cautious consumers of any science, really. But certainly of fMRI. Okay? So some features of it. It's expensive. Okay? $1 million we talked about. 5 to 7 per hour. And we'll contrast that to what we'll look at next about EEG, which is really cheap. It might cost $50,000 to start up, and about $10 for a 3-hour session. But EEG won't let you see any structural information. Okay? The usefulness when we see pictures like this is you can get 3-dimensional deep down into the brain. That's really useful. It's just a slow picture, though, as opposed to a fast one. Right? It'd be like if you watched Usain Bolt running. Right, what's it, a 10-second race? Is that about right? You get to about 10 seconds for the 100-yard dash? Mm-hmm. It'd be like if you took a movie of him running, and there were only 10 frames, 1 picture per second. you'd see movement, right? You'd see in 1 second he'd be like this, another one he'd be like this, and another one he'd be like this. Yeah? Okay, but that wouldn't show the whole race, would it? EEG will, and we'll talk about that in a little bit. Okay? Okay, so let's see here. So, a couple things. MRI. It's been related to 5 Nobel Prize winning works. Okay, so it's very influential, very prominent. It's been a great new tool in our lifetime that's been developed. Okay? But it's been criticized at times by, say, examples of books like these as a modern phrenology. And just like we can't be seduced by gall in phrenology, we need to make sure that we don't get seduced by flashy, exciting pictures just because they're flashy, exciting pictures. There was a study done in psychology about, I want to say, 2008-ish. What they did is they tested professors, all right? And they gave them a scientific article to read about some findings. They wrote it up, and they had pictures in there, the graphs and the data, the bar graphs. You know, it kind of looked, you might have the bar graphs that look like that. You showed what was different, so you showed a little star that showed that was significant, some error bars, right? You guys seen graphs like that, right? Maybe it shows also like a scatter plot. It shows some correlations. And then they gave a different group of professors the same data, the same written manuscript, but it also included picture of a brain with a blob of really flashy, cool activity. Nothing changed in the scientific report. They added a cartoon picture of a brain because, let me remind you, this is a cartoon. It's a machine's drawing, a re-rendering of what we pulled up from those protons excited and returning, but it's still a reconstruction. All right, this is a cartoon. The professors rated the article that had this in it, the same exact data, as better research and a better paper. All right, it's just human psychology, human sociology. They found it more convincing because it had a picture of a brain. This is why I'm extolling this point. None of us are immune to this. Okay, so be careful when we're reading through things. It can be tempting as the modern phrenology. Now, Endel Tulving is a famous pioneering memory researcher up in Toronto. And he had this to say about it. He said, the single most critical piece of equipment is still the researcher's own brain. It's not the brain we're studying. The most important part of the studies is our brains. We have to know how to use our brains to interpret it right. What is badly needed now with all of these scanners whirring away is an understanding of what we are observing and seeing and measuring and wondering about. So keep it in, we need to keep ourselves in check, is what he's saying. Yeah? Okay, so let's see. That's Endel Tulving. And if you want to see more details on that, you can see paper by Wire and Holdrick. And if you want to see more readings on how to be critical consumers of scientific information and question the validity of the measures being used, upon which the interpretations hinge of any study, you can see more details about that. You can go to Dr. Joe Duarte, or Jose Duarte, as he likes to go by, has some really good tips for media in reporting scientific findings. So that's just there for your optional enjoyment if you're so inclined. Because you don't want to end up face-palming your fMRI data. Okay? So let me take a look here. How much time do we have? Oh, TMS and MEG. Let me see here. I might have to plug my computer in. Are there any questions on this, I should say, as we keep moving?
I have a question.
Yeah, hit me.
Is a PET scan good for areas of like fluid or? I ask because my mom, they keep giving her PET scans because she has a tumor, and they said it's something about fluid or where the tumor was located, because on the CAT scan they couldn't see it, but the CAT scan can see structure. So they were doing PET scan instead.
So if you know, none of the fluid, it's difficult for me to say without knowing the details as a clinician, and I'm not a clinician.
Right.
But if the question is basically, can PET scans see fluid?
Right.
I would imagine situations in which they can, if you use the right tag or the right ligand, because none of the fluid is going to be inert.
by fluid, we mean stuff that has stuff in it,
Yeah.
right? So it's not distilled water.
Right.
Distilled water has nothing in it.
Right.
Or tap water is not distilled, it has salt ions in it. That's why it will conduct electricity if you drop a toaster in it or something, right? If you drop a toaster into distilled water, nothing's going to happen, right? That is a plugged-in version. So the idea is that if you have, like, say, fluid, it's going to be comprised of a bunch of cellular materials or the things that you might be able to have some ligands that can bind to or identify.
And that can prevent like a CAT scan from seeing certain structures, or
Well,
no?
I'm not sure about that.
Yeah, I don't know either. I asked my cognitive teacher too, because we learned about
Yeah.
that in cognitive, and I don't know, it's weird.
Yeah, so I mean, so PET has basically gone out
Yeah.
of fashion for research purposes.
Okay.
But it's still, as you noted,
Yeah.
very useful for certain clinical reasons. And so, because of that, I'm basically I'm a researcher. So I don't really know
Yeah.
those details of clinical ends of things. And I would be reluctant to say anyways, given that it's about individual personal
Yeah.
medical cases. I
Right.
wouldn't want to necessarily comment without knowing the whole sort of patient workup and stuff like that. But yeah, I mean, that's one usefulness of PET is it can see other stuff that CTs can't. So, other tools that people use. One's called transcranial magnetic stimulation. Okay, this is still different ways of using magnetism, and we're going to contrast that to the observational ways that we've talked about so far. Okay, so when you talk about some of the ways of imaging the brain, once, I believe, so far, we've talked about manipulating the brain in a sort of an experimental way. What method was that? Well, today, what method?
The
Optogenetics.
Yeah.
Yeah, right? Going in experimentally, turning things on and off.
Yeah.
Talked about having to basically drill a little hole to allow a tube of light to come in to turn the light switch on and off. For rodents, you can't really do that to humans. One way that you can do that with humans, in terms of manipulating and turning areas on and off to create what we call virtual lesions that are reversible, which means it's not permanent, is something called transcranial magnetic stimulation. You'll see it abbreviated as TMS. And the idea here is that there is a direct relationship between electricity and magnetism. Okay? And if you send a magnetic pulse right by the brain, it will affect the electrical activity of the neurons right next to it. Therefore, if you send magnetic pulses to a certain area of the brain, it can disrupt the electrical activity that's allowing those neurons to work. So it's a way of turning an area on and off. It's very coarse. It's not specific very much. It cannot go very deep. But it's one way of doing that in just a temporary way. And so researchers can use that to understand the things that different areas of the brain might do or not do. Okay, we'll see some examples of that. We'll contrast that also to what we'll look at as MEG, magnetoencephalography, or just MEG. Okay, and this is not disrupting activity with magnetic stimulation, but it's now measuring magnetic fields because of that relationship between magnetism and electricity. And the idea is that if we can measure the magnetic fields and how they change over time, then it's another indirect way of walking backwards and saying, well, if the magnetic fields are changing, that must mean that some electrical activity must have happened. Make sense? Okay, so let's take a look at some of those examples. Okay, this is an electromagnetic coil. This is TMS. So you might want to affect something in this area of the head, and you'll send a magnetic pulse through this coil. Okay, and so it's non-invasive, which is good ethically. You don't have to drill a hole, but yet we can still see what happens when you turn an area sort of on or off. It can be weird and discomforting, though, for people, because it's a really strange experience to have a little section turned off. Okay, in the early days, high levels of it were involved in sort of creating seizures in some people, but now there's new sort of rules in place that don't let you get to that high of parameter stimulation. So it's nice that they're reversible and temporary. So it's a little bit better ethically than a permanent lesion, right? Yes, sir.
How's it related to ECT?
ECT, oh, electroconvulsive therapy. It's not. It's not related to ECT at all. Now, very, very different method. ECT is what you might do for,
It's just like severe depression.
yeah, so like catatonic or some deep depression, and you might try to basically send large volts of electricity to the frontal cortex. We'll talk about stuff like that related to that in a little bit. So this is an example of how you might do TMS study in a lab. Okay, you use some computers, and you can navigate it to certain areas. If you use it together with MRI to take a structural scan of that person beforehand, you can use three-dimensional sort of mapping tools to stimulate exactly that area in that human's brain. Okay? So that's kind of the idea. You can use that structural scan to guide the functional disruption of that activity. Okay? This is MEG. Okay? Now we're measuring the magnetic fields, not disrupting them. Okay? MEG is really fast temporal resolution, on the order of milliseconds. So much better than fMRI. Okay? Now, you use these sensors, okay, and they can basically detect these different patterns of magnetic fields given off by neurons when they're active or not. Okay? Some disadvantages here. It's expensive. Millions of dollars for a machine. You need to have expensive technology to basically super cool these sensors and keep them cold all the time. Just part of the gig. Okay, so it's not very common, but it can be very useful. Some other limitations. It can't go very much deeper than a little bit below the cortex in what it can detect of electromagnetic fields. Okay, so if you want to study the basal ganglia or somewhere deep into the brain, MEG might not be the tool that you should be using. fMRI or MRI, really fMRI would be a better way to see activity in those deeper structures, but you're going to sacrifice the speed of measurements in order to get the depth of measurements. And this is the opposite. Okay? You're not sacrificing the depth. In fact, actually, you are sacrificing the depth of the measurement so that you can get a better speed. Make sense? Okay, so we talked about EEG as well. This is kind of the idea here. Now you're taking it forward of really, really good really good temporal resolution. And so what it does is it measures electrical activity at the cortex. What you're sacrificing is spatial resolution. You can't detect activity much deeper than right along the cortical surface. Okay? So it's even worse spatial resolution than MEG. But it's really, really good temporal resolution, and it's very inexpensive. Okay? I have an EEG lab. That's what we do here. Because you guys don't want to pay the extra tuition fees to pay for me to get a $1.5 million MRI machine. Okay? We can do really, really good, exciting work using EEG for cheap. That tells us the information we want to know about cognition, which is the speed of its dynamics. Milliseconds, it's changing. Okay? And we can see that it changes in different magnitudes, so the volume, okay, how loud is it happening, okay, versus the frequency, okay? And we can see that it does that at different sites, at different electrodes on the head, okay? But our spatial resolution is limited to just those electrode sites. Trade-offs, okay? We can use those oscillations to see either slow oscillations like this or really fast ones. By slow, we mean slow versus fast. Okay? And we can also see that they're disrupted in cases of things like epilepsy. Okay? So you should be some normal activity, and here you can see some epileptic seizures. Okay? So, trade-offs. Complementary approaches. And then you can take that EEG activity, and you can also look at what's called ERP activity, which is the averaged electrical potentials. Also very near and dear to my heart. So instead of looking at raw EEG traces like this, you look at the average of hundreds of those traces, just using that subtraction logic we talked about, okay? And that's what we're looking at here. You might present a sound. That's your stimulus. Maybe while this little guy is taking a little experiment about how he hears things, for example, okay? and you might get this raw EEG trace, but you measure that EEG trace for hundreds of trials, you average them all together, you get this nice smooth looking wave, and the idea is that you've averaged out the background noise, and what remains is the signal, just like under subtraction logic. Okay? It can be very useful. Okay? You can also take this method, EEG, intracranially at times. So just like it has really horrible spatial resolution, because you can't tell anything beneath that top little layer of the cortex that's emanating that electrical signal, in rare instances of when people are engaging in neurosurgery, like we've talked about with Sir Charles Sharrington and mapping out certain areas of the brain prior to surgical removal of them, it offers researchers the opportunity during that mapping period to place electrodes on this skull. Now this is what we're looking at here. This is a surgical prep. You can place a grid of EEG electrodes right on there. You can ask them to do different tasks because they're awake, incredibly enough. Okay, because how many pain receptors are there?
0.
Yeah. Okay. And that's basically what you can do. You drill that in, and you can also engage in like deep brain stimulation. Okay, so you can basically inject or stimulate different areas of the brain through other invasive methods. Okay, your spatial resolution can be very good, but it can also it's because it's very precise, but it also doesn't get any bigger areas. Okay, if you want to know about big networks of neurons, how they communicate, you don't want one electrode dialed into a really specific area because it's not going to tell you anything about the network. EEG might be good for that because it measures the activity of millions of neurons. Yes?
So this is called the IEEG, correct?
Yeah, just because it's intracranial. That's what the I stands for. But the technology is fundamentally the same. You're just taking an electrode and measuring electrical activity of the brain. Okay? And we can drop those electrodes into these depth sites, say in the medial temporal lobe, and you can see that the different brain areas, okay, this, for example, this is the rhinal cortex, this is going to be that purple area here, versus the hippocampus, which is going to be this red area here. Okay, these different electrodes, say on this needle, are going to record different patterns of activity in one area versus the other, and they'll differ over time as well. Okay? Make sense? Really useful. So now we can use that to study cognition and see that these different brain areas are active at different times in different ways when we remember and forget. Pretty cool? We'll talk more about that later. But this is just to introduce some examples of the method. Okay? We can also use direct electrical recording in other species. Okay? You can see it in rodents. You can do it in monkeys. Okay? While you present something on the screen, they can give a behavioral response, and you can measure that neural activity of that area's activity. Okay? So it's a very sort of basic idea here. That's what you might show. They might fixate in the middle, and you might show them something to attend to, and say this top right-hand side of the computer screen. And you can measure the electrical activity in, say, the visual cortex, and these plots are going to represent spikes of brain activity. We'll talk about that when we get into action potentials and stuff next week. Okay? So these are ways to measure single cells as opposed to large communities at the scalp, all with electricity. Okay? Now, the opposite side of that spectrum, okay, is to measure no activity at all, but to measure behavior. All right? We can see how behavior changes based upon damage to different areas. Okay? What we call lesion studies or neuropsychological approaches. Okay? So you can go in and you can cut out areas like Florenz did or like Lashley did in animals. Okay? So you can have surgical ablation in animals, see what results. We can also observe deficits in humans like we talked about with Broca's patients, with Wernicke's patients, with Phineas Gage. Okay? You're observing the behavior that goes along with patients who might have large or small areas of damage. Okay? Or in disease populations like Alzheimer's. Now, these are not reversible lesions, like TMS, all right? We won't talk about cooling so much in this class. They're permanent. Some of the disadvantages here is you don't have a lot of subjects. You have a small sample size oftentimes. Also, you don't know what things looked like before the damage. So you're very limited in the conclusions that you can draw about what deficits are related to what damage, because maybe they were not very good at that task before the injury or disease. All right, so some trade-offs. Okay. Okay, so we talked about some of those disadvantages. It can be small patient size, so they can be rare. You have some ethics involved with working with patient populations. You need to treat them as a vulnerable population, which puts a lot of constraints and requiring very careful attention as researchers to not take advantage of them and their disabilities. Okay, you don't know necessarily. You can't control where the lesions happen. You might get one patient with damage here, another patient the damage might come down here too. Every patient's going to have a lot of variance in that, so you can't control that. That makes it tough as a researcher to draw meaningful conclusions. Okay? A lot of times, if you're sick from something that gave them a disease or an injury, they can have other comorbidities. They can have. So for example, I do neuropsychological research on patients with hippocampal damage that they get from going without oxygen because they had a heart attack. Well, most people who have heart attacks are middle-aged, and they tend to have heart attacks because they have pretty unhealthy lifestyles. Okay? Some have comorbidities in that I don't know for sure necessarily, unless I'm very, very careful in who I pick to go in the experiment, that results might not be due to hypertension or to depression or anxiety or, other comorbid things that tend to go along with an unhealthy lifestyle. Okay? But they can still be very powerful. How many pigs do you need to see flying through the air to know that pigs fly? Huh? One, right? For a while, people didn't know that there was a thing as black swans. They thought all swans were white. But all it took was finding one swan that was black to know that it is possible, in principle, to have a black swan. So they can be very powerful approaches, too. What's a caveat to that? What if you find a really cool, crazy result in one patient?
Doesn't mean it's duplicatable.
Well, it might not be replicatable.
Right.
Replicable, yes.
You can't generalize it to the entire population.
Well, yes. I would say, though, that you might not be able to generalize it to the entire population, but you might be able to. Because it can be very powerful, right? So this black swan effect or the pigs flying effect. Right? Yes, you might not be able to replicate and see another pig flying, but if you saw that one and you're really sure that it was right, it doesn't mean that they can't necessarily. But what you would like to do is kind of see it again.
Yeah.
I want to make sure you're not crazy, which is one of the caveats here, right? Just because you see it in one patient or one instance, right? Maybe it's just wacky. Maybe it's just a crazy thing. How many of you guys have? Maybe that person is just weird. As a subject, like how many of you guys, right? We all know like that kind of like one weird friend or family member that's just kind of wacky, right? How many people know somebody like that? If you're not holding your hand, you're probably that person in your group of friends. Okay? Maybe that was the person you ended up testing. So, strengths and weaknesses of these approaches. Okay? And this is kind of represented here. So you can take a look at this later at home. You're measuring our time scale, temporal resolution, versus spatial resolution. Okay, and we can see where these different measures fit along that spectrum. Because every method is going to have a compromise of space versus time. And the best approach for us to draw conclusions with is across different studies that use different approaches to space and time measurements. Right, this idea of convergent validity. Okay, so this is here to sort of summarize that. And this is a chart that you can fill in your own study. Here's a bunch of methods. Describe them. What is their discrimination in time or temporal resolution versus what is their spatial resolution? And of course, all under the caveat that it's relative. Right? But this will give you that relative idea. Okay? So from that point, I'm going to take one deep breath in and out, come up for air, and dive back down and talk about some of the related ethical issues with some of these methods, or things more broadly about the science to keep in mind as we embark on our scientific journey for the rest of the quarter. Okay? Before we do that, are there questions about the previous material? You guys are locked and loaded, ready to go? All right. No? Working on it? What do we got to work on to get after? We got to like do some jumpy jacks or something? Do some push-ups to get the blood flowing? Yeah? No? Okay. All right. All right. Refresh. pivoting to sort of the ethical issues to consider in science, because no science is perfect, okay? Just because we read something that says, scientists say they found something, right? Well, guess what? I got a lot of friends who are scientists, all right? Me and my friends, we talk a lot of bull at the bar. Scientists are not necessarily any bigger harbingers of truth than any of the rest of us. Sometimes we're right, sometimes we're wrong. And it's important to be able to delineate those times, which is why I sent some of those mentions about understanding fMRI better, making quality responsible deductions, and being able to deduce about the conclusions that were drawn. Okay? So scientists say, you can read this little article, it's just sort of a paper, and if you're interested on your own, but they're basically saying science is an epic competition of ideas, players. These players hurl arguments and counter-arguments back and forth for years as a consensus maybe slowly emerges. Okay? Assessing the state of the game from any one fragment is like trying to plot a weather system from the erratic motions of a single snowflake tumbling through the wind. All right? This guy's saying, yet legions of reporters about science are trying to do just that. Tell us what's happening in the weather by looking at one snowflake, because this one study found this, and scientists said in this study. Okay? So, let's take it. I did one Google search before class. I think I just included the word scientists say. Okay? And it turns out scientists say that your mind isn't confined to your brain or even your body. Oh, that's fancy. Scientists said it, must be true, right? How about scientists say that starting work before 10 a.m. is similar to torture. You guys are laughing like you agree with it. Maybe it is kind of true, huh? That's why they put the class at noon. Yeah.
My favorite one is that people refocus on Facebook all the time.
It's got to be true.
That if you're lazy or you have like a messy room, it just means you want to tell it.
Yes. Yeah, so scientists said they did a study, and they found a correlation between people who have a messy room and intelligence, right? So it must mean that, right? So scientists said in the paper, be careful of this crap, okay? It's not modern. We've talked about it with Aristotle. We've talked about it with Gall. This is not a new story. And no, we are not immune to it because we're smarter than everyone else. Because we're not, and we're not immune. Okay? Aristotle thought the brain was a radiator. Yeah, if you don't know what that is, make sure you go back and check our notes because we talked about that. Okay? Oh, here's a good one. Overdone French fries, toast, could increase cancer risk. Scientists say. I'm not a cancer scientist. Maybe it's true. But be careful to jump to that conclusion based on one study. Okay? Live forever. Scientists say they'll extend life well beyond 120 years. Maybe? Maybe not. But just because scientists say it don't necessarily make it true. So I want to talk a little bit about, as sort of the rest of the class, about some of these elements of experimental design, some of these elements of how researchers should conduct themselves, and oftentimes do not. Okay, so that we can be valid scientific consumers and be intelligent about what we're doing. Okay, so we talked about this at the beginning of class, first day. Science is constantly moving. Heraclitus said you cannot put your toe in the same river twice. The water is constantly moving, and it is always new water. Okay, so science that we have is tentative. It's not a thing. It's a process. And we work on that towards it by using theories and models. So the theories, okay, are broad frameworks that integrate many observations from different angles to try to come up with an explanation for a complex sort of event or system. Okay? A theory, a good theory, there's lots of bad theories, a good one will generate hypotheses, but specifically generate hypotheses that can be tested. If all you do is generate a hypothesis and you can't test it, you've generated crap, or at least something that's not useful. It's not useful, I tend to call it crap. I think that's a fair thing. Okay, so if your theory is that drugs that decrease dopamine activity will improve functioning in schizophrenics, that's something you can test by manipulating drug intervention approaches with dopamine and measuring the effects of effectiveness on, say, schizophrenic measures of cognition and behavior. And that can derive from a theory that integrates observations. So emergency room physicians were observing lots of different observations that people kept getting admitted to ERs exhibiting traits like schizophrenia. However, they were under the influence of methamphetamines that were making them look schizophrenic and act schizophrenic. Well, we know what methamphetamines do. They're going to affect and overactivate certain dopamine systems in the brain. Therefore, well, gee, we have all these diverse observations. Maybe we can create a framework by which to understand that maybe schizophrenia is related to that chemical that we know is activated, overdone in these other people who exhibit the same types of behaviors. Make sense? So, and we can generate a theory that tests that. That's an example of how we're moving forward, okay? So we talked about correlational studies beforehand, okay? You can have a simple observation that A is correlated to B, but it doesn't mean that it causes it, because there could be a third variable involved. Okay, and we've talked about these approaches already. This is just a review. Already talked about somatic and behavioral intervention, ways to actually come in with experimental approaches to see if you manipulate the variable, will it change the dependent measure. Okay? So this is just a framework. This is just an overview. You don't have to memorize this. We're just reminding what we've talked about before. Okay? We've had these correlational approaches. This slide has been seen before. It's from your book. Okay? Reminding we talked about correlations, right? Between how smart you are and how messy Rose's room is on Facebook, right? You're not that smart, or the room's not that messy?
I didn't repost it.
Oh, okay. So, right? Correlation is not causality, okay? So, we talked about you can look at positive correlations, you can talk about negative correlations. Okay, this is a reminder, okay? Because a lot of times when we see scientists say they've mistaken these reminders, or they've forgotten these reminders about correlation, okay? So if you see that there's a strong correlation between two variables, let's say as the PTSD gets larger, okay, or PTSD in small brain volume, okay, what's something that you must consider? That if there is a relationship between how severe PTSD is and the size of a particular brain structure, in this case the hippocampus, right? Well, there's a correlation. What can you deduce? Is it basically that, awesome, now we can publish this new discovery saying that PTSD causes brains to shrink?
No.
Hell no. How about, sweet, stop the presses, tell everybody small brain volume actually causes PTSD?
No.
No?
No.
No. What can you deduce?
There's a relation, it's not a cause. It's
Yep.
relational.
Yep. There's an association, not causative, okay? All you can say is chillax, everybody. Correlation is not causation, okay? So, how about this correlation they ran with in Scientific American? Does sex make you rich? Okay? So, somebody said on their Saturday night Twitter feed, accelerated to a totally non-sensationalistic Gawker article called More Bang for Your Buck. People who have more sex make the most money. Well, sometimes not even that, yeah. Right? There's no causative relationship here, okay? Now, Gawker's no longer in business, but maybe this is one of the reasons why, okay? Bad science reporting, okay? We want to be responsible scientific consumers when we interpret our data and when we report it, okay? So here's some more examples that are fun. Okay, you can go to a website, tylervigian.com, and he gives you a bunch of spurious correlations. You can type in different factors and see how they're all related. For example, there is a significant correlation between the number of people who drowned by falling into a swimming pool with the number of films that Nicolas Cage appeared in. That's real. This is science, right? I mean, do you believe in science or do you not believe in science? Right? These are the absurd questions people get asked. You either believe in science or you don't. No. It's just a correlation. Lots of things are correlated, like this. Okay? How about the number of pirates? It's correlated with global temperatures. Right? That doesn't mean pirates cause temperature change. Right? But correlations exist. How about how much cheese is consumed per capita is significantly, mathematically significantly correlated with the number of people who died by becoming tangled in their bed sheets. Okay? All sorts of absurdities. Right? This is fun. In the Wall Street Journal, okay, they had a paper talk about how you could predict the Super Bowl, or excuse me, predict the stock market performance for that year based on who wins the Super Bowl. Right? It's an absurd idea. But it was just associations, okay? This was a paper that they really had, okay? Based on the different calls, okay? It doesn't mean causality. So lots of fun examples here. You can go to those sites if you want to check out the slides and play around with the different things. You can pick and see what's related to cheese consumption or whatnot, what have you, okay? But they're big issues because a lot of brain science is correlation. All we just talked about with fMRI. That's all just correlation. What activity is correlated with a checkerboard on the screen? That doesn't mean the checkerboard causes that activity necessarily. It might, but we can't say it as scientists because we don't know for sure. Okay? So we need to be ethical in how we're working on the science and how we're interpreting it and how we're sharing it with people because they're going to look to you guys back home as the people who know what you're talking about brain science. We need to be responsible when we share it too, okay? So it's important not just to be ethical in how we interpret it, but also how it's collected and how we treat each other in the process. So how do we know it's ethical, all right? Maybe something that's in fashion, like phrenology. If it's popular, is it ethical? Probably not. What about if it's conducted by trained doctors at least? That's probably a good start, right? Probably helps to make it more ethical, especially if it's supervised by like university officials, and they agree upon it and stuff like that. But it turns out none of this holds water. Okay, we can look at examples. Just because it's doctors who might be doing something doesn't make them any more or less ethical than anybody else. We have profound examples of that going back into Francis Gall as well, right? So, how about something that's in fashion, conducted by trained doctors in clinical settings? Well, this is a frontal lobotomy. This is very popular.
What the heck?
Yeah, what the heck exactly? This is a treatment for psychopathologies in the first half of the 1900s. Okay, it was spearheaded by Dr. Walter Freeman. He'd go around and he'd show this to emergency rooms and hospitals all over the country to treat psychiatric patients. They couldn't figure out what was wrong with them. What people that we might think of today that might have autism and a host of other disorders. Okay? Well, the guy in the white lab coat is walking around, he's really telling people, and he's popular, he's published, well published, so people respect his opinion as a doctor. Other doctors respect his opinion. Okay? And you get the rest of the room of doctors, oh yeah, we're going to help, we're going to hold this person down. And what you do is, you take this, and the idea was that there was a disruption in the frontal cortex. And so what you do, if you disrupt the frontal cortex, you disrupt that dysfunction. It'll help the person out. So you take this rod, and you basically come under their eyelids, through their eye socket, you jam it in here, and then you move it around a little bit. And you just screw everything up.
Really?
Oh,
Yeah, really, it's crazy.
It's a thing. It is a thing. This is a few-minute conversation about research ethics. You can do a whole course on Walter Freeman. Google it.
Crazy.
Search out frontal lobotomies because this is what you do. You take that stick and you move it around in there, and you see lesions like this that are developed. Okay. Prominent medical science at the time, predominating opinion, conducted by doctors in white lab coats. The science was settled on this. For 50 years, this worked. The science was settled. Like 9% of scientists agreed, or doctors were like, this was what to do. Okay? So prominent, in fact, this wasn't just something you did in the back hospital rooms of, underserved populations that didn't have people advocating for them. Okay? It's very prominent. Perhaps, who knows the most famous frontal lobotomy patient? Probably. Yeah, Rosemary Kennedy, JFK's sister.
Crazy.
Their father, Joe Kennedy, decided to do that procedure, probably because she might have had what we might consider today as autism. She wasn't behaving the way he wanted her to. Or psychiatrically, there seemed to be some offness, so to speak. so they did. This is a beautiful young girl. Became institutionalized the rest of her life. Okay? Very prominent. You know, there's no bones about this. Very famous. You know, it's Ted Kennedy. It's JFK Jr. Okay? This is her later. Okay? So it's not something that's immune to one section of society or class or wealth or anything. It's just there are things to be aware of. Okay? Ethics are important. And it's important to constantly be reassessing that from outside points of view. Okay? So, some of them seem easy and obvious, right? There's also gray areas, right? People love to have good Facebook conversations about the gray areas of what's difficult. Ooh, this is so, right? And, you know, but the best part about a lot of this science right now is that we have established principles and examples, okay? There's pros and cons, but we've already basically established a lot of guidance. A lot of that comes from famous studies that generated the field's interest. You know, the Stanford prison study, hopefully, from your earlier classes, okay? But we have NIH standards now, okay? So if any of you guys are interested in those details, these are links to the official sort of United States ethics that are supposed to guide our science, okay? We have institutional review boards, okay? So these areas that you think are gray, I'll challenge you back with at times to say they're not so gray. We actually have rules that guide most of those situations at this point, okay? But when in doubt, follow them. But also, just follow the rules even when you're not in doubt, okay? You're engaged in this research, okay? So we have that. We'll talk about some examples of that. You know, basically a bunch of examples, sort of like frontal lobotomies of cases when people royally screwed it up, okay? So, for example, some gray areas that are contentious, very difficult to resolve sometimes, right? How about stem cell injections to treat certain psychiatric diseases, such as Parkinson's? Okay, well, what you can do is identify that Parkinson's is due to a deficiency in cells that make dopamine. So what if we can inject some stem cells that are engineered to grow and produce dopamine? Great idea. It works. Some of the drawbacks, though, is what happens when cells grow to make dopamine cells, right? They keep growing. Well, when cells continue to grow uncontrollably, that's what we call cancer. So when they have done experimental treatments like this, they've literally given people cancer at times, right? Puts some pretty serious boundaries ethically on some of the scientific approaches that we can do to treat some of these conditions, experimentally and also clinically, okay? So a very difficult sort of balance to strike, okay? When we do that with humans, one of the guiding principles is we have informed consent, okay? Which means they're engaging voluntarily and you explain everything you can beforehand. As an example I had to deal with. I was a graduate student. I put my research subject in an MRI machine. He was crying because he was a 6'4" college football player and we were studying recovery function from concussions. Well, he thought he was in pain and he thought he wasn't going to be able to play football anymore. He was a scholarship. He was crying. He didn't want to be in there. And he's 6'4" in those tiny little MRI boards. Okay? I called my supervisor at the time. I said, yeah, this is what's happening. He's like, well, just talk to him, see if he can see if he'll just finish the experiment. No bueno. That's not NIH compliant with our standards of research conduct. All right? Because it violates the informed consent that says they can cease or stop participating at any time. Right? Which goes all the way back to that Stanford prison study. Okay? So we talked about that. Sometimes you can use deception, but you want to follow some very clear rules when you're doing research on that with humans. The milligram shock experiment, hopefully you guys are familiar with as an example from your intro classes for that. Okay, so there's clear boundaries and ethical guidelines that guide that. Speaking of ethical lapses in informed consent, there was a prominent study from my alma mater, nonetheless, UC Davis. I was shocked to see this. There was a neurosurgeon who ended up, thankfully, getting fired because he did some experimental treatments on brain cancer patients while they were dying and never got institutional review board approval. Well, what was the experiment that he was doing? The idea was that if you could only get the body's immune system to attack the cancer in the brain, that it will beat the cancer and destroy it. So as a way to lure the immune system to attack the cancer on the brain cancer patients, he put fecal bacteria on it to lure the immune system approach. Okay, you can check it out. I mean, you know, it's a very interesting study, or interesting write-up. There was a theory that he had that guided it, which is fine, but it wasn't institutional review board approved. Several of the patients died, which is still dicey, because they were going to die anyway. I mean, we're all going to die anyways, but these are some very complicated issues that help to guide the research that must be abided by. Okay, so some other stuff. Plagiarism is the theft of another's work or ideas. Something that's very prominent as an issue that's still important in the field. You've probably heard it since 5th grade, right? Well, there's a reason we're talking about it in college right now. Okay, because it doesn't go away as an issue. It's actually quite prominent. Okay, so it's purposely taking or due to carelessness or even lack of knowledge, it still counts, and it's theft of another person's work, of their ideas. All right, it's not something that successful people are immune to. Okay, and if top leaders weren't doing it, then we wouldn't be talking about it. Okay, now, along with that comes another issue of scientific ethics: fabrication. All right, some people scientifically have been caught straight up just faking it. All right, I'll share some examples with you. It's pretty crazy. Okay, but basically the storyline is don't do these things. Okay, they're bad. Okay, it gives everybody in science a bad name. Okay, it's a big issue in social psychology right now. There's big scandals that have occurred on data fabrication that has allowed people in society to question the entire validity of the field of social psychology because it had been so prominent. Okay, so let's see. Talk about some examples. Okay, so what do all these people have in common? Dr. Martin Luther King, Vice President Joe Biden, and Fareed Zakaria. Any ideas? Yes. Yes, all plagiarists. Famously and provably so. Okay, this is from the New York Times. A Boston University panel found that Dr. King plagiarized his doctoral dissertation. And then they kind of looked the other way and were like, it probably wasn't so bad. It was bad. Don't do that. Okay? Fareed Zakaria got basically suspended from CNN because he was a bald-faced plagiarizer on multiple occasions. Okay? Biden canceled his 1988 presidential campaign with a bunch of egg on his face. You probably can't see it, but there was egg all over this. Because he got caught plagiarizing a bunch of speeches. And his thesis, I think, in law school. Anyways, the idea, though, is that we're not immune to this. It might sound like a very simple thing, like kind of like reiterating. No, like this goes up to the top, which means that we're all, need to be reminded of this stuff. Okay? How about data fabrication? Anybody know who Michael LaCour is? Okay, this is a fun case study. Okay, Michael LaCour is a graduate student at UCLA.
Oh no.
Oh yeah. Okay, so here's what happened with Michael LaCour. Okay, he ran a study about gay marriage. It got published in the journal Science, the biggest journal on the planet. Big, prominent finding. It turned out that if you canvassed and went to people's house directly, this is back a few years ago when there was a big issue of gay marriage in society about changing views on marriage, right? And the storyline went that people were kind of set in their views. But he reported this exciting finding that if you had people of that orientation actually do the canvassing and talk to people face to face, that actually changed attitudes, which is a big finding. Especially had political implications, social implications, and they ran with it. They loved it. They put it all over the media and the news and guided politics and law and everything. Well, it turns out people thought it was so amazing that they wanted to do studies like it too, and they kept tracing it and tracing it and tracing it, and they couldn't figure out why they couldn't replicate it. And it turns out once they saw his data, it was all fabricated. He made it up out of thin air, and he got a job at Princeton.
Oh my gosh.
Yeah. And so you can read the whole story. I put links to all this stuff down here. It was basically another gay researcher at Stanford who thought it was such a cool finding he wanted to build upon it. And he had basically kept trying to do it, and he had to blow the whistle. And he published papers like, guys, this is total crap. He faked it. So it's very prominent. This is that scientific hero, as you might think, the guy who blew the whistle on it at Stanford, and it was a very, very interesting scientific story about lapses in ethics.
Wait,
Okay?
how was he able to find out that data was saved?
Because he ended up getting a copy of it. And he could see where it was added, and the data was too clean. When you talk about stats in your stats classes, you'll find that good data has variance in it. If it's too good to be true, it is. And so this social psychologist also found guilty of fabrication. Okay? The cool, if you really want to have fun and follow a lot of this stuff, retractionwatch.com is a place where you can check that out. And stuff gets retracted all the time, usually for perfectly benign reasons. You know, accidents happen, you know. But occasionally there's nefarious stories that make for fun conversations in class. Okay? Let's see here. So examples of that in psychology as well. How many of you guys have heard of the IAT, Implicit Attitudes Test? Yeah, really famous, prominent study. So in New York Times, they just had a thing about a month ago, actually, chronicling the ethical lapses of how that test is being used to suggest things that the designers of the tests at Harvard and Columbia admit that it cannot actually do. Okay? Scientific ethics guide us to walk ourselves back from making claims that the data are not supported by. Okay? So you can check that out if you're interested in some fun reading for that. Okay, so I'm going to skip this part about patient HM. Remember, there are reporting mechanisms for things, and to know the rules, follow the rules, basically don't do the wrong thing, do the right thing if the rules aren't followed, and let's see here. I'll kind of wrap it up there. I guess we have, what, 2 or 3 minutes? Is that right? Yeah?
3.
Okay, so stop putting your stuff away. Let's talk for 3 minutes. You guys got a test coming up, right? All right, cool. If you're staying on top of things and following the syllabus supplement that I suggested on how to study, following the regular syllabus suggestions on how to study, what to do, you guys are going to do great. You're going to do fine. But dial in. Get good sleep. Get good rest. Get good nutrition. Don't cram like crazy and come totally strung out and underslept. You're not going to help yourself. Okay? There's 3 big things. I'll mention them again on Thursday that always trip people up. The best students in classes have always come to me and they say these things after the test, who I know are good students. And I know they know it. And they go, oh, I missed this one because of this. Oh, I can't believe I got this one. There's these 3 things. Just give me these 3 rules I now suggest. Okay? Which is 1, read the question. We read it too quick and we miss 1 or 2 words. Changes the whole ballgame. Slow down. Read the question. 2, read the answer options. The best students, and right, I've done this too. We've all screwed up these little. I go, oh, I forgot that I didn't read it all the way through. I rushed through it and I thought it was this. No, slow down. Read the options carefully. The words are going to matter. 3, just answer the question. Right? You don't have to overthink it. Well, what about all these other things? Da, 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 da. I'm just giving you a heads up. Try to just shoot you straight on the test. We're just nice and easy. It's not an easy test, but not trying to have trick questions. So just think about what it's like being a witness on the stand in a crime movie. All you have to answer to the attorney grilling you is yes or no. All you have to answer is that question. They don't want to know what you think about everything else. Real simple. Make sense? Those 3 things catch people the most more than anything. Because I know you're working hard. I know you're studying. Keep it up. You'll do great. Just be careful not to rush. Just be patient in walking through each one. Yes?
You have the whole class, right?
Yes, you have the whole class. But like I was saying earlier, people tend to not need it because we save a lot of time by not having to bubble things in. Yes, ma'am?
I haven't read chapter 4 yet. Did you get to cover it all today? Or was it like
Well, we covered the good chunk of 4 today. We didn't cover much of it in the past.
so when I'm reading, do I like stop where we stopped on the slides? Pretty much?
Yeah, I'd be careful to say, me in terms of like, not like sort of liability-wise, I don't want to tell you a specific spot to stop, but I'll tell you like topically, like I've said at the beginning of class, right? that we're not going to get into like the drug use. We're not going to get into those models of that. But I wanted you to know from chapter 4 those neurotransmitter systems that we talked about. So that's a lot of just reiterating what we talked about earlier today. Yes, ma'am.
I have 2 questions.
Yes.
How many questions will be on this?
I'm not sure yet. Haven't made it.
Okay, and the other
Probably
one is,
50.
the other one is, can we bring a tablet? Does it have to be a laptop or?
It does not have to be a laptop, but it needs to have access to Blackboard. Very simply, just any way that you want to access Blackboard and that you can. Have plenty of warning. Everybody said they have access from one mechanism or another. I will be actively walking around, keeping an eye on everyone's eyes and everyone's screens. And there are mechanisms built in place electronically, which is awesome, where they can actually detect certain features too. So just use that and be honest. Yes, ma'am.
You can just rent a laptop from the library and just bring the class.
Yeah, apparently we also have rentals available. Are there any questions? No? Okay. Sorry? I haven't made it yet. No, probably not a lot. Thank you guys. Keep up the good work. Get some rest.
It's just about axon nodes, and I feel like I understand everything, but where they are visually and how it's just right here, right? Or is it up here?
No, it's not. Where's the axon?
This guy.
Oh, okay. So you want to
Well, because in the book, like, look, this is the guy that messed me up. Because where's that connected? That's the axon.
Yeah.
Those are the nodes of yes. Right. Where is it inside of the little?
No. It just refers to it. It's that.
I'm really struggling with it.
It's the connection sort of area. It's
Like
spot
where
where that little red shade. See that little shade,
Okay.
shade? That's the node.
But it's not all throughout the whole axon,
It's not
right?
shaded.
Okay. Well, the book doesn't make it very clear because I really tried.
It's just that certain
And then
it's where things are.
well, they're having little dendrites on them. Do they have little dendrites right
You can
there?
have dendrites. No, actually, no, not.
Like these little guys, look.
Yeah,
So
so
it's like oh, so that's the axon pillar. This is the dendrites.
those are the stones.
Okay. No, I thought for some reason I thought this whole thing was the axon.
I just gave.
Yeah.
Like seriously, because that's how people get it wrong. People are so stressed out in front of remember all this stuff and
I'm
freaking
so
out.
stressed out. Okay.
That's when message to everyone is like.
It's on video.
I'm trying.
What?
It has like a video on.
So that last third rule, I think it's just answer the question.
Is everything? I'll do my best.
No, that's what I just said at the beginning of class. Well, like I said, we're not focusing on like the illicit drug classes.
I was wondering why they introduced these two terms here on this page. Do you think this has anything to do with it or just put in there? I understand this side in the cell body. Why would you introduce the two here?
Just to show that
Is-
the systems are not just anatomically different systems, but they're chemically different systems.
So which one is neurogenic? Is it this side or this side? Or which one is the core?
Sympathetic is the
Okay, so this, right. So the sympathetic is this one and the neurogenic is this one. Okay, all right, so it doesn't really say that. I mean, it's there. He had to put it there for a reason. I'm trying to figure out why. You know? So this side will be the sympathetic, and then this side, the neurogenic, will be the parasympathetic. Okay, all right, I'm just making sure. I didn't know how to use that information.
Okay.
Okay. Okay. Well, just don't look at other people's things. Why don't you wear a watch? If you wear a watch, then you won't have to. But yeah, I mean, I appreciate you letting me know. Yeah. Well, I mean, you should not be turning around and looking backwards. You should be walking backwards. Me too. I mean, that's okay. I understand.
Thank you.
You're okay.
It is.
Thank you, sir.
Yeah, I think you're a good gift. You know, I think you'll like it because it's research. It's not opinion. And I think you're going to really like it. The best book I can find, but that's for somebody else, so don't worry about it. But other than that, but yeah, no, I really think you'll be able to use it. I didn't tell you, but I mean, I could. Senior Vice President for the Los Angeles Times, I challenged her. She didn't believe the science. She took me into it was about 15 years ago. She took me in, it was a $100 challenge. I met her and she didn't believe me. She took me into the executive dining room, which they had at the time, which they don't have. And I sat in there and I sat with her. And we were there for over the lunch, a 2-hour period. I said, I picked out 3. She said, this is what LA. This is, we don't have the dress code. We don't have this in. And in 3 hours, I picked out 3 people that walked in. All 3 of them were above her. She's a senior vice president. All 3 of them were above her. I said, those 3 people are just dressed correctly. I never seen these people before. All 3 of them are above her. Nobody else walked in the room that was better, was dressed properly. They were all below her. She was a vice president. And after that, she believed.
I believe it.
And then she hired me.
Fantastic.
Yeah.
Thanks, man.
So it works.